New topic: genomics. The term genome was introduced by H. Winkler in 1920. Definition: the total genetic constitution of an organism or complete copy of genetic information, that is DNA. Term genomics coined by T.H. Roderick in 1986. The study of genomes through analysis, sequencing, and mapping of genes along with the study of their function. The types of genomics: structural genomics, it involves mapping, sequencing, and analysis of genome. Functional genomics, it involves study of functions of all gene sequences and their expression in organism. Then application of genomics: number one, structural and functional genomics used in improvement of crop, plant, human health, and livestock. Second, the knowledge and understanding can be applied in medicine, biotechnology, and social science. It is used to develop transgenic crops of desirable characters. Then, lastly, genetic markers have applications in forensic analysis. Then, Human Genome Project. It was initiated in 1990 and completed in 2003. Aims of HGP: mapping the entire human genome at the level of nucleotide sequences, to store information collected from the project in database, to develop tools and technique for analysis of the data, transfer of related technologies to the private sectors like industries, taking care of legal, ethical, and social issues arises from project. DNA fingerprinting: every individual has its unique genetic makeup called its fingerprint. The technique developed to identify a person with the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. This technique was developed by Dr. Alex Jeffrey in 1984. It is based on identification of nucleotide sequences present in DNA. About 99.9% nucleotide sequences in all person is same. Unusual sequences of 20 to 100 base pairs are repeated several times, called variable number of tandem repeats or VNTRs. Length of the regions having VNTR is different in each individual, hence they are key factor in profiling. Steps involved in DNA fingerprinting: number one, isolation. DNA must be recovered or isolate from small cell or tissue of body, for example blood, hair, skin, nails, etc. Restriction digestion: the isolated DNA is treated with restriction enzyme. This enzyme cut DNA into small fragment having variable length. This phenomenon is called restriction fragment length polymorphism, RFLP. Then gel electrophoresis: in this step DNA samples are loaded on agar gel electrophoresis. Negatively charged DNA fragments move to positive pole according to their length. This results in formation of bands. Southern blotting: separated DNA fragments are transferred to nylon membrane or nitrocellulose filter paper. Selection of DNA probe: a known sequence of single-stranded DNA is prepared, called DNA probe. The DNA probe is labeled with radioactive isotopes. Hybridization: in this step probe DNA added to nitrocellulose filter paper to form complementary base sequences. The next, photography: nitrocellulose filter paper is photographed on X-ray film by autoradiography. Then application of DNA fingerprinting: number one, it is used in forensic science to solve rape and murder cases. Second, it is used to find out biological father or mother in case of disputed parental. Then it is used in pedigree analysis in cats, dogs, horses, and humans. Let's watch the given video to understand the concept of DNA fingerprinting.
DNA fingerprinting is a method of identification based on an individual's DNA. Now let us understand the process in detail. In the process of DNA fingerprinting, the tissues such as blood, skin, saliva of two different individuals are taken, from which the DNA is isolated. This isolated DNA is then cut into small fragments with the help of restriction enzymes like restriction endonucleases, which generates various DNA fragments of different sizes. The fragments are then separated on the basis of size using gel electrophoresis. The separated DNA fragments are then transferred or blotted to synthetic membranes such as nitrocellulose or nylon. Radio-labeled probe molecules are then added that are complementary to the sequences in the genome. These probes contain repeat sequences, which show very high degree of polymorphism and are called variable number of tandem repeats or VNTRS. The probe molecules hybridize to DNA fragments and the excess probe molecules are washed away. The hybridized DNA fragments are detected by using autoradiography. Radioactivity from the probes produce dark bands that corresponds the position of VNTRs. As seen in the image, the two samples A and B have different pattern of bands showing that they belong to two different individuals.
This way, we complete our entire chapter, molecular basis of inheritance. Thank you.